Hello, my name is Lucas. I have a Bachelor of Science in Physics, and I'm going to present my undergrad research work that I did in the supervision of Professor Fonseca and Lucas Araujo. So, for a quick introduction, the use of radiation has become very important in the last few decades, but we have seen that the use of high doses of radiation can pose some risks. So, the main dose of radiotherapy, for example, is to deliver the highest dose possible to the tumor while keeping the patient as safe as possible, delivering the lowest doses possible to sane tissues all around. So, radiosensitization has become a way to kind of improve treatment safety and efficiency. Well, and nanoparticles are an example of these radiosensitizers. What they do is basically, if a radiation beam, an electromagnetic radiation beam, hits a nanoparticle, it is re-emitted as a lower reach radiation, which is electrons, and those are limited to the area of the tumor, so it kind of doesn't let that pass to sane tissues, increasing treatment efficiency. So if you want to study that, you would need, if you want to study that computationally, you would need models, computational models. And there is today a huge difference when you compare macroscopic human models to microscopic models. As you can see here in the poster, we have a cell model used in 2020. It is basically just a cell. It is a sphere. The nucleus is a sphere, and the cytoplasm is also a sphere. It is very simplified. So the goal of this study is to present two distinct image processing methods that will help create better microscopic models for computer simulations. So for case study 1, materials and methods, we have used as a cell model HeLa cells. They have, like you can see in the poster, the dimensions of 350 micrometers by 350 micrometers, and they are comprised of many images. Like in the Z direction, there are many images, so 129. Each of them represent 1 micrometer in depth. The images, they are only of nuclei of HeLa cells, and they were binarized by the authors that made them available to us. So what we wanted to do is kind of individualize to analyze the effects of radiosensitization in a single cell scale. So what we had to do was to individualize these cells. We used a Python script based on connected component algorithm to identify these cells and individualize them, so you can simulate the radiosensitization effect in only a single cell. We also did nanoparticle distribution simulation, so we can analyze the effects of nanoparticle radiosensitization in it. For case study 2, we have used osteosarcoma cells instead. They are fluorescence microscopy images, so we can see here they are comprised of a red channel, a blue channel, and a green channel. We wanted to voxelize them, make models out of them, but separately, we wanted to separate the different structures, so we have voxelized individually the red channel, blue channel, and green channel. So for that, we used Python and Vedo for creating those objects, and we voxelized them using Benvox and Benvox Merge, which are programs that were created by a programmer from the Netherlands. And we have converted these Benvox files to MCNP inputs using a program that was developed by Professor Telma, which is called Benvox to MCNP. So for the results in case study 1, we have seen that all discretization of these nuclei, it was really successful, because we have used like another plugin from ImageSay to validate our results. We have seen that the differences between our results and theirs was of around like 0.001% in the average difference of the coordinates of the center of mass of each nuclei in any direction. So it was only a very small difference was found when comparing to another tool. And you can see in the poster that we have successfully simulated nanoparticle distribution. We have distributed it in four main ways, like outside the nuclei in clusters, randomly in the medium and on the nuclei surface, and inside the nuclei. So for case study 2, you can see here that we have successfully created a model. This is already the 3D input file, as you can see. And yeah, the voxelization and conversion was successful. So this study introduced two image processing methods for creating 3D microscopic models to support investigations into radiosensitization effects of nanoparticles in biological tissues. The scripts presented in this article will be made available as image processing tools in the future. Thanks for listening.